Thank you very much. Thank you for the introduction, and thank you very much to the organizers for this meeting and for having me here. It's a huge honor and a real pleasure to be at the place where basically everything started, right, in fly vision science. And so I'm, oops, this is rolling away. I'm mostly interested in how the visual system interacts with active movements or animals, or maybe the other way around, how active movements interact with visual processing. And both for the case where flies move through the environment because they want to go from point A to point B, which generates optic flow that they somehow have to deal with, but also how animals move the sensors actively to shape the sensory input they have access to, and how they then in the end benefit from that. And you all know that many animals move their antennae, whiskers, fingertips, ears, and also eyes. And those are just two very well-studied examples: microsaccades in a primate, and the optokinetic reflex in a zebrafish larvae. And when you look at this stationary scene, your eyes start from one fixation position to another, and at each fixation position, you're going to perform these microsaccades. And in this classic example by Yarbus, you can basically infer which parts of the visual scene the human observer has been most interested in. So eye movements are very widespread across the animal kingdom. They come with functions, but they also have this problem that whenever the eye moves, it generates motion on the retina that the brain somehow has to disambiguate. And in fact, while you are performing these large saccadic movements, you're not aware of the motion those eye movements generate, because your brain somehow blinds you to the consequences of your own saccades. And I'm interested, or my lab now is interested in the mechanisms by which this is achieved, and the visual benefits of eye movements on the flip side. And in my postdoc in Gabi Mehmenstedt at Rockefeller, I was working on a mechanism that is closely related to this stable visual percept in humans. And we were asking how flies can momentarily ignore unwanted self-induced motion. And so people who work in flies know that flies have very strong stability reflexes. So basically, if you show visual motion to the right, to a fly, the visual system will show a response. It will trigger a motor response that turns the fly to the right. And they move their wings to turn to the right, they walk, they move their head. But then on the other side, you know that animals don't only move in response to acute external stimulation, but they also move spontaneously. And so if you have a fly in a stationary environment performing a voluntary left turn, this will have a visual consequence, which is rightward motion on the retina, which then would trigger exactly the same visual response and would, in the absence of any modulations, trigger optomotor turn that is opposed to the spontaneous voluntary left turn. And this has been termed reflex trap. So animals that have very strong stability reflexes need to shut down momentarily those stability reflexes to be able to move spontaneously or voluntarily. And it has been proposed in the 50s that there could be a way to escape this reflex trap by sending an efference copy with every efference to the visual system to cancel out the visual consequences of a turn. And funnily enough, this has at least similar ideas have been put forward by Sperry, for example, but those two von Holst and Niedelstedt came up with this idea by looking at walking flies. And then many years later, it was possible to record from the visual system in behaving animals. And we were looking for a signature of such an efference copy. And so the neurons that we picked are the so-called horizontal and vertical system cells. So flies have a simple brain, so to say, but it still consists of more than 100,000 neurons, and 60% of those are somehow involved in visual processing. And we picked this particular set of neurons because they have been long known to respond to panoramic visual motion. So they respond to horizontal visual motion and vertical visual motion around different axes. And as an ensemble, they are very well poised to quote or to tell the fly in the movement it performs through space. And so they are also probably the most well-studied neurons in any invertebrate. And ever since the discovery, they have been implicated in gaze stability reflexes. And so first, now with the genetic tools in Drosophila, we wanted to check if we can silence those neurons and inhibit gaze stability reflexes. And to do that, we put flies on a little metal rod in a visual arena. And you're going to be able to see head movements, but not wing movements, because the frame rate of the camera is off. But I hope that you'll be able to see that they move their eyes with the visual motion. You can basically make them dance with these little movements. And then we did the same experiments in flies where we inactivated HSV cells using an inward rectifying potassium channel. And we saw that this head optomotor response is clearly reduced compared to control genotypes. The wing response was very much less affected, maybe in the case of the fastest motion, but at least those neurons seem to be important for head movements. And so then we thought, okay, let's think about how flies move, and this is complicated, but the main takeaway is that we think that the main visual consequence for a flight turn, which is a banked turn, is yaw visual motion. And so we wanted to measure the visual responses of this ensemble of neurons in response to this yaw visual motion that flies experience during a typical flight turn. And we performed wholesale patch clamp recordings, and here I'm just plotting the average response to, for example, rightward motion. And as expected, horizontal system cells respond strongly, but you see that also vertical system cells show responses to a varying degree. And then we wanted to know what happens in those neurons when flies perform a flight turn. And I'm going to show you how this actually looks like. And this is done by Anmo, who was my close collaborator in the Meman lab. And so this is the view that we have of the flies when we perform those patch clamp recordings. So they are tethered, rigidly tethered, so that we can perform electrophysiology, but we see the wings from below, and we can indicate the leading edge of the wing beat amplitude. And the difference between the left and right wing beat amplitude gives us a measure of the attempted flight turn direction. And so you see here a time series of such a wing beat amplitude trace, and this downward deflections would be what we think leftward turns. And then we can at the same time record in wholesale patch clamp recordings, in this case in a horizontal north cell. And here you see in the absence of any acute stimulation, there's this coinciding hyperpolarizing input that comes with this wing flicks. And this is going to happen again and again, and then she's going to stop saccading, and at some point she's going to saccade in the other direction. So here now we have rightward turns, and then we get depolarizing input. And we quantified the amplitude of those motor-related potentials for the set of visual neurons. And when we do that, for here one saccade direction, we see that the amplitude of those motor-related potentials is opposite in sign to the visual, the amplitude is matched to the visual response, and the sign goes in the opposite direction. So we think that if the fly were free to move, the visual response of the neurons and the motor-related input would cancel each other out. And we think that this is a quite sophisticated mechanism that sends quantitative cell-specific efference copy to momentarily disengage the stability reflexes. Okay, and we have later found that this also happens when you scare flies with looms. So if you try to swat a fly in your kitchen and they escape in this beautiful way, a part of it is this mechanism that unfolds in their visual system. And so we really wanted to know where this efference copy signals originate from, where in the brain do they come from, how are they distributed so nicely to those different visual neurons. And this would have been my follow-up project in the Mehman lab. But before I was diving into that, we paused and we were asking if it's not perhaps possible that flies, like so many other animals, also move their eyes. And this is important for that question because eye movements would then potentially come with its own set of efference copy, and to fully understand the system, we would have to know that. Classically, this is not how one thinks about insect eyes, right? So insect eyes are often thought of being stationary, but it's also clear that at least some arthropods move their eyes. And I did my PhD in Vienna in spider psychophysics, and I used as a readout the activity of the motor system of the eyes. So spiders can track motion with their retinas. And from that time, as a PhD student, I was very much aware that arthropods move their eyes, and that and also of the literature. So here there are spider and human eyes, the invertebrate eyes and the vertebrate eyes that we know that move are at least superficially quite similar. So they have a cornea, a lens apparatus, and the retina behind. Insect eyes, on the other hand, have this, as you all know, hundreds of individual ommatidia and lenses that are rigidly connected to the carapax. But the literature that I was just mentioning that was known is that Roland Hengstenberg in 1971 found this muscle that attaches inside the housefly to the frontal part of the retina. And he suggested that this muscle is poised to twitch the photoreceptors underneath the lenses. And then 20 years later, Nicola Franceschini found a second muscle, and Franceschini and colleagues published a series of conference proceedings where they showed that actually those muscles move the photoreceptors underneath the lenses in 2 dimensions. But then, interestingly, in Drosophila, no one really followed up on that. And so it was unclear whether Drosophila has those muscles, and if so, how they would use them. I'm sorry. And so we first wanted to check if Drosophila has the muscles, and they do. So this is a stack that Igor Sivanovic, my collaborator in Janilia Farm, provided. And you see here 2 muscles stained with haloidin in red, attaching to this cuticular ridge. And this is how Igor imagines the 3-dimensional arrangement of those muscles. And here in yellow is the orbital ridge at the base of the photoreceptors. And so we said, like, okay, this is interesting. Drosophila and other flies have muscles. What would they be good for? And many eye movements in the animal kingdom have the purpose of shifting a restricted field of view around, or a specialized area on the retina, like the fovea, for example. But flies, and in particular Drosophila, basically see the entire visual surround with a very homogeneous resolution. So the question is, why would you move the eyes, right? And the ideas we got from the literature is that eye movements in flies could, for example, help to counteract photoreceptor adaptations. If you have a completely stationary eye in a completely stationary environment, the percept fades out over time, and you could imagine that twitching the photoreceptors would bring the percept back. Maybe more excitingly, eye movements could potentially help to increase the spatial details that animals have access to. So again, if you imagine a stationary eye, the spatial detail you can resolve is harshly limited by the angle between the receptor units. But if you can move them over time, you can increase the spatial detail you have access to. And actually, engineers make use of jittering sensors to do exactly that. Then third, you could move the eye to stabilize moving objects, and potentially eye movements could also provide depth cues. And so we really wanted to know if to what extent Drosophila moves the eyes, and if we can see eye movements. And the first try to investigate this was to just move the head of a fly in our normal holders that we use for patch clamp recordings, so that we can see basically through through water immersion onto the lenses. And because the refractive indices of water and biological materials are so similar, you can basically see through the lens. And here I'm showing you a focal plane up here, and you saw some tiny jitter, which mostly comes from the fly flying on this tether. And I'm gonna zoom further down, and I hope that you'll be able to see whitish grape-like structures that are moving around, and those are the tips of the photoreceptors. And you see that the sheet of the retina is twitching. And so there's dirt on this slide, and it's not a perfect example, but this was basically the only time I tried this in this setup, because it's nice and easy, but then it gets hard if you want to stimulate the eye that you are observing. And in order to observe eye movements in a more realistic or experimentally accessible setup, we made use of the deep pseudopupil that was actually also figured out by Nicola Franceschini and Kuno Kirschfeld. And the deep pseudopupil is a quirk of the compound eye, which is basically an enlarged virtual image of the photoreceptor tips inside the fly eye. And it's quite easy to see, so it's sufficient to shine red light through the head of the fly and point a normal air objective onto the eye, and you'll see this deep pseudopupil. And if you're so inclined, you can count the individual photoreceptor cells. And the thought is that if the head is rigidly tethered and does not move, but this pseudopupil moves around, it tells us that the photoreceptors themselves have moved. And because we know the spacing of the photoreceptors, we can roughly calibrate how much they have moved in angular space. And so we built this little setup where, again, we have flies on a metal rod flying. We can monitor wing movements, and we pointed 2 cameras onto those on the 2 different eyes. And at the same time, we can show visual stimuli. And when we did that, we saw very regular movement that coincided with this grating motion. And for now, I just wanted to focus on the fact that the head of the fly is not moving at all. It's just the retinas that are moving. And what we are doing is easier when we zoom in. And so here I'm highlighting the centroid of the right and left pseudopupil, and I'm going to plot the horizontal excursion over time down here. And you'll see that whenever the grating moves to the right, the pseudopupil moves to the left, and vice versa. And if you go through all the signs, those movements slow down the retinal motion, so the relative motion on the retina, and they're therefore consistent with an optokinetic reflex. So very similar to the example in the zebrafish that I have shown you in the beginning. And it's a very reliable reflex. So basically you see this in any fly that you put in this arena. Unlike humans, they can do this completely independently in both eyes. So if you only stimulate one eye, only one eye moves and the other one doesn't budge. And this is very impressive in the average. And they can also do convergent movements and divergent movements, and they can also track up-downward motion, but to a smaller degree. When you blow on the fly, so this is all data from a fly that was just hanging on the stick, and if you gently blow on her, she's going to start flying, and then everything gets more lively, so you get more counter-saccades that sometimes really look like nystagmosaccades in the human optokinetic reflex, but overall they still do the same thing. They still track the retinal motion, at least in these slow phases. And we then wanted to know, do they also move the eye spontaneously? Right, and they also do that. So if you here have a fly flying in a stationary arena, you see these small jumps happening. Although the flies can keep the retina very stable for extended periods of times. I'm gonna switch off the lights, and then you see that there are much larger and more frequent saccades happening. I'm recovering from a cold and have not entirely recovered. Okay, and when we switch the grating on again, then she goes immediately back to this strategy of stable positions that are interspersed by small jumps. And those are really small. So if you plot the XY trajectories here, you see that the stable positions are separated by less than 1 degree. So this is a fifth or sometimes a tenth of the interomatedial angle in flies. And we think that potentially those movements could help to refresh the image or to sample space in finer detail. And this is something that we are following up on in the lab right now. But also interestingly, at least superficially, they look like a little bit like the microsaccades in the monkey that I have shown you initially. And I think it's going to be interesting to understand why an animal that has such different eyes and brain attached to the eyes perform strategies that at least superficially look similar. But when we found those little jumps in the beginning, we were concerned that they would have any effect on the visual system at all because they're so small. And to address that, we recorded again in horizontal and vertical system size, this time just because we know so much about those neurons and it's easier to interpret any findings. And so here, you see a very classical response of a horizontal system cell to rightward and leftward motion, depolarizing for one motion direction and hyperpolarizing for the other motion direction. And it got really interesting when we looked at those neurons in the context of a stationary environment. And what you would expect here would be a flat line, right? There should be a stable baseline membrane potential. And actually, perhaps I would have thrown away that recording a long time ago because there's no stable baseline. But now that we know that there are eye movements, we can plot the retinal position together with this membrane voltage, and we see that these relatively large depolarizations, so that's the same recording in the same cell, so those are quite substantial depolarizations, those coincide with the small retinal positions. And to quantify that, we picked moments of small retinal jumps and averaged that over animals and then over the population of flies, and could ask what happens at those moments to the membrane voltage. And we indeed saw a depolarization for one motion direction and a hyperpolarization for another motion direction. And if you go through all the signs, those responses, the large responses here, are consistent with visual responses. And indeed, when you switch off the lights in the same cells, those responses are entirely gone. You might be able to see that there's if you squint, there's this little quiggle at the onset, and we think this is a small motor-related potential that grows when the flies are flying. And then we were surprised. Those are small movements, 0.5 degrees, and we get a direction selective response. It's in a way shocking. And we wanted to know if we have calibrated our eye movements correctly. And to address that, we recorded from so-called LC14 neurons that have a relatively constricted field of view and have basically tile the frontal part of the visual field. And we were asking if the receptive field shifts when we move the retina, and instead of measuring a two-dimensional receptive field, we just measured the response to a stripe because the main excursions in yaw. And we moved the muscle by activating the motor neuron by expressing CS crimson, a redshifted channel rhodopsin. And whenever we shine red light on the motor neuron, we can shift the retina and then hold it in place. And this is how the traces of the eye look like when we quantify it. And you can see that we have nicely separated positions, which is very handy if you want to measure the receptive fields. And we can at the same time perform whole cell patch clamp recordings in those neurons. And you see that at least first pass, the visual response is intact. So even though we shine red light on the fly, and which are not very sensitive to, but a little bit, and we pull the retina to quite some degree, the visual responses are okay. But then we can zoom in further and ask whether they shift, right? And so those are averaged example traces in one fly for right and left but stripe motion. And then this is the same cell when we optogenetically activate the muscle. And you see here a shift of 15 degrees, which is really a lot, and it's kind of surprising that the visual system still functions, right, under those circumstances. And we did this for a population of flies, and if you then average, this shift in receptive field very nicely corresponds to the shift that we calculate from the pseudopupil movement. So to summarize that, we found spontaneous and stimulus-triggered retinal movements. Some of the movements are tiny, but still impact the visual system, and others are relatively large. So 10 degrees is something that flies do in actual natural behavior. And then we wanted to ask what the function of those retinal movements would be. And here I'm showing you experiments that were actually the first experiments done in my own lab in Martinsried by the first master student, Lukas Rand. And we were looking at gap crossing behavior inspired by the work from the Strauss lab. And this work has shown that flies can judge the distance of a gap and change the gap crossing behavior accordingly. So they can launch themselves over it or walk around. Flies, at least Drosophila, don't have a large binocular overlap, and they have relatively bad resolution. And so it's unlikely that they could use stereopsis in a way that we use stereopsis. They also don't perform very obvious peering movements when they approach this gap. And so it's unclear to what extent motion parallax helps. And so we thought maybe this is a place where retinal movements could play a role. And so we basically copied the setup, and you're going to see flies crossing a challenging gap. So this is 3.5 millimeters, which is roughly a body length. And this fly is going to launch over, and the other ones are going to walk around. And this is the fastest you'll see. And so we then thought, okay, let's silence retinal movements and see if anything in the behavior or in the gap crossing strategies changes. And we did this by using two separate Gulf-4 driver lines that both target these retinal motor neurons. They're quite sparse, and you basically probably can't see anything, which was a problem for us because the expression levels are not very strong. So we could not get fully rid of eye movements, but we could strongly dampen them. And so we had flies with dampened eye movement cross those gaps. We tracked their trajectories using the bleb cut. And then in example traces, you see that a control fly, for example, here was crossing all the time on top. So this would be the depth of the gap and the distance they have to cross, versus the fly with the silenced eye movements showed a more variable crossing strategy. And this held over the populations of those two genetic lines. They can still cross the gap, right? They are not blind, but they are slightly more clumsy, which is in line with perhaps a distance misjudgment. And this would fit well with what we see when we look at eye movements when flies are crossing gaps in a tethered assay, which is what we do so that we can actually see what the eyes are doing. And you see here the wheel position. So there's this wheel where we have gaps, and any gap crossing is indicated here by a vertical line. And up here you see the retinal positions, and perhaps you can see even just looking at this example trace that whenever the fly cross a gap, there's a large eye movement happening. And this holds over this one fly and over the population of flies. And this is a convergent eye movement. And this convergent eye movement, we think, might provide flies with a dynamic depth cue that would only work really in the very, very front and just at the range of a body length. And I'm happy to discuss later how we think this potentially might work. And we are now bringing that setup under the 2-photon to look if we can see or dissect the algorithms by which neurons make use of this shifting receptive fields to judge depth. And so in summary, we found this retinal movements that at least in some circumstances look like vertebrate movements. And we now want to make use of the tools we have in Drosophila to understand all the axes of this active sensing model. So we want to understand the motor commands, where in the brain is it decided when and in which direction the eye should be moving. And for that, we make use of the electron microscope reconstruction of the fly brain. We want to understand if the efference copy mechanisms, so is the visual system informed about an impending eye movements, and then we want to understand the perceptual benefits. So why do flies even do that? And what do they see because they move the eyes, and how is this implemented in the brain? And my lab is really young, but I'm gonna show you the first basically insights that we gained. So I have one PhD student who is working on this efference copy problem, and so she's recording from lobular plate tangential cells while the fly is moving its eyes, which would come with a changing visual input, a visual consequence. And she wants to find out if the efference copy or perhaps other modulations like proprioceptive feedback. And she, this is Tialda. Those are the flies with the deep pseudopupil and the lobular plate tangential cells. And this is a recording, for example, of a HSN cell in darkness. And again, you should have expected a flat baseline. But what we see these fluctuations that at least qualitatively seem to correlate with retinal position changes. And the sign of those fluctuations is consistent with efference copy signals. So they basically would oppose the consequence of a visual of a retinal movement. And Tialda is trying to figure out where this all comes from. So what's the circuit mechanism? And then for the perceptual benefits, I just told you we are making use of all those Drosophila tools and 2-photon and imaging neurons to understand how perhaps they do depth perception or have hyper acuity. But the first project that was actually launched in my lab to that question took a more comparative approach. And we wanted to see if we can connect retinal masses to visual ecology of different insects. So when we started out, we just knew those 3 flies have retinal movements. No one knew about dragonflies, beetles, etc. And so a PhD student in the lab is looking at eye movements across insects. And he has also perfected imaging techniques to visualize these minute muscles in insects ranging from tiny squishy ones to large pigmented ones. And surprisingly, after basically sampling 25 out of these 29 insect orders, it seems that retinal movements have evolved very late. So in the Mecoptera, Diptera ancestry. So those are Mecoptera scorpion flies and Diptera true flies. And within Diptera, we see a very interesting variability from 0 to 3 retinal muscles. In some species, it's actually dimorphic. And we want to sample this now more densely to then connect it to visual ecology like fast flight, foraging behavior, visual courtship, etc. And with that, I would like to thank you for listening. Gabi Mehman and the Mehman Lab, in which lots of the work has been done. The people who launched my lab, basically, Igor from Janilia, the fly resources, and the funding.